Continuing on with these different components that are inside our body and our cells that help fight off infection. Now, complements, or sometimes known as complement proteins, there's about 20 different blood proteins that are circulating throughout your body. They're kept in the inactive form because if they were activated at all times, you could risk increased destruction of your cells. Now, they didn't really have very original naming schemes here. We're looking at C1, C2, all up to C9, B, D, and P factors. It's just very basic, simplistic numbering and lettering of the names. But this really is going to be one of the major factors playing a role in getting rid of foreign particles in your body. So getting rid of those pathogens, those viruses, bacteria, whatever's in there. This is a major player, major function in how your body does that processing. So pretty much the complement is going to amplify all aspects of your inflammatory process, as well as pretty much helping to kill off bacteria. You can kind of think of this almost as a power-up to the inflammation process, a power-up to your immune system. It's really helping to amplify what is going on. The key thing though is, what's nice, is it also increases not just that non-specific, like the inflammation, but also your specific defenses get boosted. So it's helping to boost both the innate and the adaptive defenses. So that allows it to really be a very essential component of your immune system. So now when we look at this, we can kind of see this is what's going on. Now you notice there's a lot of different things, a lot of different parts and pieces. We don't need to memorize every single one of these factors. That's beyond our scope. But a quick overview, and I'm not going to blow this up because I don't really need you to know which one's C3 versus C5. But the key thing is, in the beginning, you're going to have the initial complex proteins binding to the antibody. So this allows those first ones, C1, 2, and C4, to kind of bind initially, split up, and start this whole cascade. As you go through the cascade, it's bringing and attracting phagocytotic cells. That's going to be important because it helps to destroy things. As you keep going down this cascade, they're breaking apart, bringing different enzymes in, kind of going through the whole process. Ultimately, here's the goal. By the time you get to the bottom here, the end result of all these different proteins and different parts working together, they're all going to kind of come into and drill a hole in that pathogen. Now, hopefully remember back to AP1, talking about the membranes and how they're regulating internal environments, because you're trying to regulate your different ions and liquids. If you get a hole drilled into a bacteria, do you think there's any way of regulating inside and outside? No, that's it. It's done. So at that point, they drill that hole in, that cell is going to die. It'll basically break apart, lysis, and you're done. So this complement protein not only can amplify the whole process, it can go through this cascade and destroy a cell as well. So a lot of different parts and pieces to work through here. We don't need to know which one is C3 versus C5 versus C2. We're not worried about that. Understand the cascade of these proteins allows the ultimate result of destroying by drilling a hole and creating this open space for fluids and ions to pass through. Inflammatory response. Now, I'm willing to bet you're saying, yeah, I know what that is. I've dealt with that. I've had injuries. Yeah, fine, sure, you probably do know. But the inflammatory response is any time tissues are injured or infected. Now, from our purposes, we're talking more about infections, but injuries can have the same result. The whole goal here is to try and contain, prevent widespread problems. So it doesn't matter if it's an injury or an infection, you're trying to contain that point and make sure it's not causing more problems. So in the case of an infection, you kind of get rid of those pathogens, get rid of the cell debris, the broken components that are being left in the wake of that pathogen. You're trying to get yourself set up and ready for repair. Because if you cut your skin, for example, pathogens get in, inflammation occurs. You're trying to contain everything at that point of where the breach occurred. Trying to get back to the point of healing and seal it back up again. So a few things that you'll typically see for inflammation: redness, heat, swelling. You're going to sometimes have pain with it. Actually, most of the times you have pain with it. I'm willing to bet you've probably seen that. You've probably experienced that. Now, sometimes, though, you also get impairment of movement. That's where if the injury is at a joint, maybe you sprained a ligament in your elbow, or you tore the ACL, they'll start to kind of swell and kind of make things a little bit harder to move sometimes. So you still get the redness, the pain, the swelling, the heat, because that's all that blood. Everything's rushing there to make that work. That's inflammation. But these macrophages, as well as the epithelial cells of boundary tissues, they can have these things referred to as toll-like receptors, TLRs, toll-like receptors. Now the toll-like receptors, they're going to basically check and see what's being infected. So they have very specific classifications of different microbes they can respond to. So they make sure that they have an idea of what's coming in there. So it recognizes each class, each different division of these infections. So then you activate these TLRs, these toll-like receptors. They release cytokinins. Cytokinins promote inflammation. So you're starting this process off. These toll-like receptors are one of the initial steps that will occur in the inflammation process, as well as in your body's response to how you deal with infection. Because they're being released immediately right when that first break, that first rupture of the skin occurs. So you're trying to make sure that your body's getting prepped and ready to defend. Now you have an injury here. Nail hits the skin. Basically, it could be anything, just as long as the skin is ruptured. That breaking of the skin releases the toll-like receptors. They can initiate their process. First thing occurring is leukocytosis. Leukocytosis is the increasing of white blood cells to an area. In this case, we're looking at the neutrophils, because neutrophils are the primary response unit. You'll have more neutrophils than anything else responding. Others will respond, but not in the same numbers. Then you have margination. What that means is the white blood cells are kind of flattening against the walls in this area. You're saying, well, why would you flatten against the walls? That leads up to diapedesis. Diapedesis is the white blood cells kind of squeezing out of the blood capillaries. They're going into the actual infected tissue. And then they follow the path of chemotaxis. Chemotaxis is going to be a chemical trail leading to the bacteria. Because just like any invader, any chemical, any process here, everything leads a trail. Whether it's your white blood cell traveling through here, whether it's a pathogen, they have little chemical trails or signatures they can follow. So chemotaxis is your blood cell following that trail to the pathogen. Once it gets there, destroys it. Phagocytosis, or depending on what cell it is, could be marking it for any of the other processes we've talked about. All right, this one can look kind of daunting because there's so many boxes here. So we're going to take this and blow it up full screen. Starting off up top with tissue injury. Now, this is going to go down a couple different pathways. The first pathway we're looking at is release of chemical mediators. Think things like histamines, prostaglandins, looking at complement kinins, the chemicals that start the whole process. So we look at vasodilation of arterioles. Well, if you vasodilate an arteriole, that means you're increasing blood flow to the area. That's like local hyperemia. By increasing the blood flow to that area, you bring more blood in, which means you have more redness because there's more blood, and you increase the temperature. Because blood's coming from the core of your body out to the extremities, it then increases the temperature. That local increased temperature helps to increase metabolic rate, leading to the healing process. Next one up here we see is increased capillary permeability. By increasing the capillary permeability, you're increasing the leaking of fluids out of those capillaries. Leaking the fluids out allows this protein-rich fluid to get into the tissues. Well, you need those proteins because the proteins are what are there to help rebuild the tissues. because your tissues are built out of proteins. So you need them to rebuild. But this can also lead to swelling and sometimes pain, because as you have all this fluid leaking out, it's causing the swelling, and your tissues can only swell and move so much. Now, this swelling could potentially lead to temporary limitations of joints. So obviously, if the swelling is not in a joint, you're not leading to limitations. But if the swelling is in a joint, it could have an impact on the range of motion, or simply cause a complete limitation of any motion at all. But again, the ultimate goal, lead to healing. But that leaked fluid, that's where the lymphatic comes in here. Your lymphatic system has to pick up that leaked fluid. So we're having an increased flow into the lymphatics, trying to help balance out the amount of swelling. So all this fluid's leaking out of your capillaries for the healing process, but at the same time, your lymphatics are picking up all this leaked fluid. So it's trying to balance between the two, make sure you have enough of that protein-rich fluid to help the healing process, but also not to cause too much swelling. Third pathway here you see is attracting neutrophils, myocytes, and lymphocytes. This is all via chemotaxis. They're all trying to come in, follow the pathways of infection, and get rid of the foreign pathogens. But if you notice also that one that popped up underneath that is that leaking of fluids. So you're also getting clotting proteins here to help. Get these fibrin patches that are helping to block fluid loss, block blood loss. So it's creating this kind of structurally built environment to help aid the healing process. And then the last one going down there is seeing the release of leukocytosis-inducing factors. Basically, leukocytosis is the increasing of white blood cells in the area. White blood cells are phagocytotic, amongst other things. So therefore, you have this increase in the area. You have the marginalization occurring, we just mentioned. That margination is getting ready for diapedesis. So that's literally was the same exact thing we talked about on the previous slide. The whole point, clear the area of debris, which means all of these are trying to get back to the healing process. So that shows that we have a whole lot of things occurring on here, but they're all having one goal. They're all trying to heal the damaged tissue. Get rid of the pathogens, get rid of cell debris, bring in things that can help to heal, help to build, help to recreate, as well as helping prevent fluid loss and allow everything to regrow. So a lot of things occurring, kind of going back to some previous chapters to cover things there and bring it into this one for the fluids being picked up from the healing process. Which brings us into fever now. Now fever, I'm willing to bet you probably have an idea what it is, because most people probably had a fever at this point. Fevers are going to basically be a systemic response. We're talking head to foot. It doesn't have a fever for your arm or your head. The whole body gets a fever in response to something invading your body. So what's happening here? We have leukocytes, we have macrophages. They're all going through and secreting pyrogens. Now, these exposed pyrogens, they're going to help with resetting the temperature. They're also activating these leukocytes, activating these macrophages. So it's helping to kind of kick your body's immune system into a higher gear. But those pyrogens, while they're present, will maintain that fever. So your body's at a normal temperature, and everyone's body is a slightly different normal temperature, but wherever your normal is, that's where your body stays. The reason when you have a fever, you stay at the higher temperature is these pyrogens. They reset your normal thermostat. They say, hey, this is the normal. This is where you should be. Trying to get your body to work at that heat, work at that temperature. So a whole different barrage of ways your body can fight off foreign infections. From fever to complement proteins to interferons to you name it, there's a ton of different parts and pieces here. All these chemicals, all these cells are meant to help your body fight off foreign pathogens.